So we've talked about chromosome segregation and cell division so far, and we've done a very reproducible process, the process that really allowed us to go from one cell to all 30 trillion cells in our body, mostly, and that's the process of mitosis. But we're going to finish here just by briefly talking about the process of meiosis and how that's different. So this is also a cell division event, but it's one that we want to use instead of making two equivalent daughter cells. We're going to use this to make the gametes. So in our case, for example, we're going to use this to make sperm and egg. In the case of yeast, you're going to make spores, things that can survive. And the key thing between these and most of the cells in our body is that these gametes are going to be haploid. So most of the cells in our body are diploid. There's 2 copies of each chromosome. And here, these haploid cells are going to have 1 copy of each chromosome. So we have 46 chromosomes in our cells because we got 1 pair from our mothers, 1 pair from our fathers. And in this case, we want to be able to generate cells that can reconstitute that. So an oocyte's going to have 23 chromosomes in it. A sperm is going to have 23 chromosomes. And when they come back together, we'll be able to fix this. Okay? So I want to take our chromosomes again. Now I have 2. These are the 2 homologs. Okay? This is chromosome 1, but it's from your mom. This is chromosome 1, but it's from your dad. Okay? And so we have these chromosomes here. And ultimately, our goal is to make sure that every cell gets just 1 of these. Okay? And so you could imagine doing that, where I do a first division exactly like we've been talking about. I pull these 2 apart. I end up with 2 different ones over here. I separately do this for those. But when I try to do it again, when I try to create a division that allows me to fully distinguish them, and just one over here, one over here, one over here, one over there, we could ultimately end up with 4 different cells, which you can in the case of yeast, for example. All 4 chromosome products will end up in a spore, and it's called a tetrad. Okay? And so each of those is a haploid cell with 1 chromosome of each that you began with. Okay? So we still need to replicate the DNA, but we need to come up with some way to distribute it a little bit better. And so we have this wonderful trick that we have cohesin. I've solved that problem for this chromosome during mitosis. I can do that. I've solved that for this one. I need these chromosomes to work together. I need them to know that they exist and that they're going to work together, and that way they're going to distribute appropriately. Okay? And so in addition to the cohesin, I need a way to make sure that I can glue these together in some way. Okay? So as we'll refer to a diagram on the board in a second, we're going to begin by really actually just pairing them, having them find and recognize each other and line up. And so we're going to build a structure called the synaptonemal complex, which is going to hold these together. But then I'm still back in where I am, and that I can't actually figure out how to separate these in any sort of logical and coherent way. Okay? And so then we're going to actually use a second trick. So these chromosomes are really nicely intact, but I'm going to have a way to put them together in some way. And so actually, I'm going to do something that you would never want to do to a chromosome, which is I'm going to break it. Okay? So I've broken this chromosome here. It's got a piece. I'm going to break this chromosome too. And I'm going to create a situation where I can allow these chromosomes to mix and match. Okay? So normally I wouldn't want to be able to do this. But here I'm going to break them in such a way that I'm going to allow them to refuse and recombine with each other in different ways. Okay? And so now I've created a different kind of structure. These are going to be these recombined chromosomes. Let's see if I can do this well. And so now they're actually all held together. So even when I get rid of that synaptonemal complex structure, all these chromosomes are held. And it kind of looks like a mess. But now we're going to do some different things to be able to separate stuff. Okay? So I still want to do a cell division event. Okay? I have the recombined chromosomes here. And they're held onto each other. So I replicated them, I paired them, I held it together in exactly the same way we wanted, but then I swapped the DNA across just at one place. Okay? So we're going to divide these chromosomes, but instead of doing that the way that we've been doing thus far, we're going to release the cohesin, but we're going to hold on to it at centromeres. So any cohesin near the centromeres I'm not going to cut, but in this first meiotic division I'm going to come through, I'm going to release all the cohesin along the arms, but I'm going to protect it exclusively at centromeres. And I'm going to do that up here too. And so in this case, again, that cohesin goes away, that cohesin goes away. And in doing that, I'm now going to release these two from each other. And so these, during this first meiotic division, these are the things that are going to segregate. Okay? This one's going to go this way, this one's going to go that way. So I've lined them up and paired them, but just by cutting the arm cohesin, now I've released these from each other. Then I'm going to come back through a second division. Now I actually need to be able to distribute these. And I'm going to go through and I'm going to cut that centromere cohesin. And I'm going to end up with these two new chromatids. But these chromatids are going to look, still chromosome 1, still in exactly the same way. But what you should see here is that at least one of these chromosomes has a part from the dad chromosome instead. So if these were the two mother chromosomes, all of this region here came from the father. Okay? And so this process of recombination will help resort our genomes. And so if you imagine, your siblings are going to look different even though you came from the same source material, if you both had the same parents. And in this way, we can actually resort the material over evolution within a population. But what you really need here is this ability to actually have this distinct cohesin and recombination to really enable the meiotic chromosome segregation.